guys, my name's Naomi. I'm a graduate student in the Kugel Lab here at the Hutch, and I'm going to show you what a day is like for us. Here in the Kugel Lab, we study one particular subtype of pancreatic cancer. It's called basal, and it's the most aggressive form of pancreatic cancer. And we're trying to figure out what markers in this cancer make it so aggressive. So one of the things I'm doing today is trying to mutate specific points in the genetic code for this marker to see how that changes the behavior of the cells. All right, so this is all to just extract DNA. And then I'm going to try and give it to cells and see what the cells do. So once I would have extracted the DNA, I'm going to run it on a gel to see if I can see the differences that I'm trying to make. So the gel will help separate the different bands of DNA, and I'll be able to tell if the change I'm trying to make is there or not. After I give this DNA to cells, I'm going to see if the protein that I'm trying to change is also being changed. In the cell, DNA, or the genetic code, is translated into proteins. And what I'm trying to do is look at if some of these proteins are high or low, because we think that some types of pancreatic cancer are really high in certain proteins and really low in other proteins. And if we know how that correlates to disease, then we can start to treat people based on those protein states. This is the final step of what's called a Western blot. It's kind of like the DNA gel I was about to run there. So here I've taken a dish of cells, and I have lifted them off the plate and broken them open to get all the proteins out from the inside. And then I run those proteins on a gel, and depending on how big the proteins are, they may be at the bottom of the gel if they're really small, or the top of the gel if they're really big. And now I have stained for those particular proteins to see if I can see if they're high or low in a particular dish that I extracted protein from. So the stain I've used will show a brown to the naked eye, but it also lets off light, and that's what this machine detects, is whether or not the sample is letting off light. So what I'm hoping to see is 3 lanes that are untreated, and those should have high levels of this protein. And then 3 lanes that are treated that should have low levels. Awesome. So if we take a look at this, you can see, so this is my ladder. That just tells me how big the proteins are. But here, like I was saying earlier, I have 3 bands that are really high in the protein because they've stained for it, and 3 bands that are really low, which means my experiment worked.